Hello, I'm John Kagan, the David and Susan Coulter head of Mechanical Engineering. Welcome to the College of Engineering's webinar series, Faculty Insights, a 20-minute briefing, and thank you for being here today. In this virtual series, faculty from across the College of Engineering share insights into their research and its impact and provide perspective for the future. Following each 20-minute presentation, attendees will have an opportunity to direct questions and comments to the speaker. Our speaker today is Vickie Webster-Wood, Associate Professor of Mechanical Engineering, and she will give a presentation entitled Biomimetic, Biohybrid, and Biodegradable Robots for a Sustainable Future. During this program, she will discuss the research she is performing in the CMU Biohybrid and Organic Robotics Group, or BORG group, building robots as models to help us understand biology and using biology as materials to build more sustainable robots. Professor Webster-Wood received her BS, MS, and PhD in mechanical engineering at Case Western Reserve University and completed her postdoctoral training as well at Case Western. Upon joining CMU, Professor Webster-Wood established the CMU Borg, where her research focuses on the use of organic materials as structures, actuators, sensors, and controllers toward the development of hybrid and organic robots and biohybrid prosthetics. Vickie is a rising star in her field and on our faculty. She leads a multi-university Murray Award from the Army Research Office and has received the NSF Career Award. She also won the Outstanding Mechanisms and Design Paper Award at IEEE ICRA, which is one of the top robotics conferences in the world. At CMU, she is the recipient of the Dean's Early Career Fellowship Award and holds courtesy appointments in biomedical engineering, materials science and engineering, and the Robotics Institute. I will also add that Vickie is an incredible colleague who contributes greatly to the community and culture in mechanical engineering and the college. Before I turn the screen over to her, I would like to remind our audience to please use the Q&A feature at the bottom of your screen to submit your questions to Vickie regarding her presentation. You may do this at any point during the program. Nikki Bernardi, Senior Director of Development for the College of Engineering, will be moderating the question and answer session with Vickie at the conclusion of the presentation. Vickie, thank you so much for being here today, and I turn it over to you.
Well, thank you all so much for joining us today. And thank you, John, again, for that wonderful introduction. Hello, everyone. It's my pleasure to be with you all for this Faculty Insights and to be able to share the work that my lab, the Biohybrid and Organic Robotics Group, has been conducting in this space of biomimetic, bio-inspired, and biodegradable robots. With our long-term goal of creating robots towards a more sustainable future. And before I dive into the actual research, I always like to take a moment and acknowledge the Borg team. The work that I'm sharing today, at this stage, I'm not the one at the bench pipetting things, building robots. This work would not be possible without an amazing crew of researchers, including my current postdocs and graduate students and many fantastic alumni. And in addition to the graduate students that have been involved in this work, it's been my pleasure to host numbers of undergraduate student researchers in the lab since its inception in 2018. In MECI in particular, about half of our undergraduate students, if not more, are involved in research in our labs at some point during their time here. And the contributions of Borg undergraduates have been integral to many of our projects. And so with credit where credit is due, let's talk about science. Animals are capable of tremendous feats of agility, strength, ingenuity, creativity, and learning. They adapt and respond to dynamic changes in their environment. They learn from experience, and they solve complex real-world problems in ways that, as an engineer, we really want to be able to reproduce those abilities in future robotic systems. Towards capturing these capabilities, in my lab, we're focused on biomimetic, biohybrid, and biodegradable robotics. We want to be able to learn fundamental principles of actuation and control by studying and modeling biological organisms. We want to then identify and characterize renewable, biocompatible materials and manufacturing techniques that we can use in our devices in order to build adaptable living actuators from muscle and energy efficient living controllers from nervous systems. And our ultimate goal with all of this work is to be able to create soft, bio-inspired, and biomimetic robots that can themselves safely interact with us and our environment. And in the future, may one day be made completely out of materials that are biocompatible and renewable. And so in today's talk, I was trying to put together these slides, and there are just too many cool things that my students are doing. So I had to kind of down select a little bit. And I wanted to share some characteristic examples of our work in these areas. I'll start out by talking a little bit about our work in biomimetic robotics as tools for basic neuroscience, and how we're translating from those experiments into more applied soft robotic applications for agriculture. And then I'll change gears and focus on some of our work building foundational tools in biohybrid robotics, in particular in the kind of design, materials, and manufacturing spaces. And finally, I'll wrap up by sharing some of our work in creating biologically sourced, marine-derived biodegradable actuators. So let's dive in. One of the foundational questions that has really driven my research vision is, how do animals generate multifunctional adaptable behavior? And this question is really instrumental in robotics because understanding the natural world allows us to ask, how can these principles from neuromechanics help robots be more adaptable? And then by building bio-inspired and biomimetic robots, we can in turn test hypotheses about the animals and learn more about the natural world. And so we create this synergistic collaboration between disciplines. But to do this, we really need a tractable animal model to get started. And by tractable, I just mean not so overly complicated that it would be impossible to model. So for this, we've been doing a lot of research studying the sea slug, Aplysia californica. And sea slugs are fascinating. Aplysia is a really robust marine sea slug with a relatively small nervous system. Where you and I have billions of neurons, the sea slugs only have a couple tens of thousands. And it's been a model animal in neuroscience for decades, which means that there's a lot of literature about the nervous system and about the muscles. From a mechanical engineering perspective, it's also incredibly interesting because these sea slugs are completely soft-bodied. It is just muscle pulling on muscle. There's no bones, so it makes for some really challenging soft body mechanics problems. And yet, from a robotics standpoint, it's still really, really interesting because with these very limited number of neurons, this limited musculature, the animal's capable of demonstrating learning and complex multifunctional behaviors. In particular, we study the feeding apparatus as a model of soft grasping. And we see kind of three multifunctional behaviors. We've got biting, which is what you're seeing in this video here. It's kind of like the mouth in the movie Alien. So the mouth comes out and tries to grab seaweed and pull it back in. If it successfully grabs seaweed, it then swallows that back into the esophagus. And if it notices that it's trying to eat something that it shouldn't, it can instead reject the food and spit it back out. And so with this animal model, we can then start digging into the mechanisms that generate this multifunctional behavior. And in this case, it starts with a single soft feeding apparatus called the buccal mass. The muscles within this structure are controlled first by the buccal ganglion, which you can kind of think of as a motor controller, and the cerebral ganglion, which provides kind of those higher level signals and processes sensory information. We can break down studying this neuromechanical system into kind of two sections. First, we need to understand what neural units are needed to model the multifunctional controller. And second, we need to understand how the musculature itself may be contributing to this multifunctional behavior through morphological computation. This starts really through collaborations with biologists and neuroscientists. And we look at the behavior of the animal, during which we can assess the key neurons and muscles that are involved. For example, in the swallowing behavior, we see kind of four phases, starting with a pinching stage where muscles are activated to hold the food at the jaws so that it can't be pushed out of the feeding apparatus. The next phase is protraction, where that internal grasper is pushed forward. It then closes or grasps on the seaweed and is retracted back into the mouth in order to ingest the seaweed. And this sequence of muscle activities generates force on the seaweed. In order to translate this into models to be able to simulate this control system, one way to think about it is that each of these bursts of neural activity can be approximated by just a Boolean burst. So it's either on or off. And that kind of can represent how the neurons are firing. And so based on our team's own prior work and existing literature, we can then build models of these biological neural networks. So in these types of maps, each of the nodes is an actual neuron that we know exists in the animal that we've simulated. And that can be connected to other neurons via these kind of inhibitory or excitatory synapses. And our neural networks, we can model them so that they can receive external sensory cues to drive behavior, and so that the motor neurons feed signals back out to a model of the biomechanics. And that can then in turn provide kind of those feedback signals that we need to generate the whole behavior. And so, then we have to start thinking about, well, how do we model the biomechanics, right? It's a very complex, soft-bodied system. When we look at this structure again, we just see, again, muscles pulling on muscles. As we've dived into this, it's really interesting. Like the activity of one muscle can completely change the mechanical advantage of a different muscle. You've got all these soft body contact mechanics going on. So it's very challenging to model in simulation. And we developed a number of models, but there's always this kind of sim to real gap that we have to think about. And so this is the first spot where robots becomes really instrumental in our research. Uh, Rather than just relying on these simulations in a computer, we can instead start building biomimetic robots of the buccal mass and get some of those contact mechanics kind of for free just through the structure of this being a physical device in the real world. So what we do is we try to match our actuators onto the actual musculature and fiber orientations that we see in the animal, and mimic those internal structures and the internal mechanics to the best of our ability with synthetic materials. We can then connect that to models of the slug brain that we can use as the controller for the robot. And then the system can receive those external sensory cues that tell it which type of behavior it should be doing, even just from a user. And in this case, this is Slugbot version 1. It feeds on this little tube of seaweed, well, tube representing seaweed, as kind of a stand-in. And we can then start comparing the behavior of the robot with our model of the nervous system to that of the animal and that of our simulations without a physical robot. And our initial simulations of the complete neuromechanics did an okay job at predicting kind of the motion of this system and the neural activities. But you're seeing that here in kind of this pink color. And when we compare that to the gray, we see that there were quite a number of differences. Without changing all that much in the controller at all, and just switching from that simulation of the biomechanics to the actual robotic model, we get a much better match between our model of the system and the actual animal data, which is showing us that we do have a certain level of understanding of how this system works because we're able to reproduce it. But there's still kind of these discrepancies in timing and amplitude. And these point towards new directions for future study to better understand how this nervous system works. And so Slugbot v2 is currently under development in the lab to address kind of these gaps that we see. But that's a lot about sea slugs, right? So how does studying sea slugs help us build better robots? Well, when we look at the elements of the slug's feeding apparatus, we see that it can be roughly broken down into these different ganglia. And we can instead think about these in engineering terms, right? So if we abstract these concepts, we start seeing things like, oh, this has viscoelastic adaptable actuators. It has an adaptable controller capable of learning. We see evidence of some time history dependent motor control, right? So we need some local motor controllers. And we've got this grasping apparatus that can close on objects. as well as change kind of the contact stiffness of that jaw region, depending on the material properties of the object that is trying to grasp. And so based on kind of these abstract principles that we see in the organism, we can then develop engineered systems that capture these features to do tasks like soft robotic grasping. And so, for an example of that, we've been developing this soft grasper that is abstractly inspired by the grasper and jaws of Aplysia. So, rather than trying to match the muscles exactly, we just use these abstract principles. And in this case, the grasper uses pneumatic actuators to safely close on the objects, and then has these pneumatic soft jaws, which allow us to tune those contact stiffnesses. We can similarly abstract principles from the control system. And we've been developing robotic controllers using an approach called synthetic nervous systems, in which we simulate individual neurons and try to capture some of the actual temporal dynamics of neurons, as well as the synapses that connect them. And then, just like in the slug brain, we saw this kind of hierarchical structure of the neurons. We can establish that same sort of hierarchical structure in our robotic controller, and then train it in simulation to do this grasping task. And excitingly, we were able then to transfer the controller with minimal retuning to the robot, which is able to likewise do this soft grasping. And it can pick up a wide variety of objects. We've done a bunch of lab objects, household objects. We've had it pick up produce, tomatoes, eggs, like you're seeing here. And more recently, some kind of work in progress that we're going to hopefully be submitting really soon. We've been doing direct head-to-head comparisons between this soft grasper and kind of your more standard robot rigid graspers. And we found that in particular, when picking up soft, fragile objects, the soft grasper causes substantially less damage than the rigid robot controller or grasper. Okay, so that kind of wraps up a look at the biomimetic branch of the lab. And let's change gears and talk a little bit more about biohybrid robotics. Unlike robots, biological systems that we're trying to mimic are really fundamentally composed of dynamic responsive materials. Our goal in our biohybrid research thrust is to be able to reliably design and fabricate robots using these living biological neurons for control and living muscles for actuation. And this all kind of falls broadly within this field of biohybrid robotics. So the idea is just that you have some part of your robot is biological and alive, and some part of your robot is synthetic. Across the field, you see this broken into a couple different spaces. For example, there's folks doing research in microorganism bots for things like targeted drug delivery. There are tissue-based biohybrid robots that often use either skeletal muscle or cardiomyocytes as the actuator. And then there's also folks doing some work in cyborg robotics, working primarily with insects or things like jellyfish. And the work in my lab all falls in this tissue-based biohybrid robotic space, where we're trying to gain the advantages of living muscle in terms of adaptability, self-healing, energy efficiency, and bring those into robotics. But even though biohybrid robotics has been seeing this growing interest over the past decade, there are still a lot of challenges that remain in translating these approaches to more complex robotic systems. In particular, my lab has been focused recently on the gap in design tools for biohybrid brains and bodies. And when we look at thinking about using living neurons for control, a big challenge is how do you get them to form the network you want them to form? So, towards really understanding how these neurons grow and ultimately being able to control that growth, we have been developing data sets and models of neuron growth towards biohybrid design tools. So we've created and shared a large annotated data set of neuron growth in culture, including quantitatively describing the neuron growth in terms of how long are branches, how many branches are there, when a branch turns, what's that typical turning dimension. And this is all really important because it forms the foundation for better neuron models. And so in work in collaboration with Dr. Jessica Zhang here at CMU, we've been developing and validating phase field models of neuron growth to allow us to predict based on cues in the environment and then the conditions of the substrate how a neuron might grow in culture. And by using our measurements from actual living neurons, we're able to get the model to better reproduce the types of structures that we see in vitro. And next up, we're working to kind of adapt this, both from a culture approach and a modeling approach, to be able to pattern specific circuits to get us the functionality we want for biohybrid control. And then what about designing the biohybrid bodies? Right. There's a lot of design tools for robotics generally, and especially for robots that are composed of rigid links. But in biohybrid robotics, we're typically working with soft, squishy, fragile components. So we've been working in this space, in particular, taking a look at the idea of biohybrid worm-like robots. And a key challenge in biohybrid robot design is that the muscles get stronger with use, right? It's real muscle. They fatigue if they're overexercised, and they can atrophy from disuse. And this is in sharp contrast to traditional robotics, where you can reasonably expect your motor won't change all that much use to use. And so, as part of this research in my lab, we've been computationally studying biohybrid designs with multiple distributed actuators towards looking at designing robots accounting for this adaptability. And the idea here is that you would have kind of this woven mesh structure, and you could pattern actuators onto the mesh. And then contracting specific actuators allows you to get these different motion primitives that you could then use for locomotion or reaching or grasping. But prior biohybrid design approaches really hadn't looked at how muscle adaptation changes the robot's ability. So to simultaneously develop a controller for our biohybrid worms and study how adaptability affects performance, we've been using reinforcement learning as both a controller and a design partner. So the idea with reinforcement learning is that you have some sort of policy or agent that can receive information from the environment and make choices about what action should be taken. For us, this environment is composed of our biohybrid meshworm. So the purple is like the structure of the mesh, the red is the actuators. And then our goal is to try to get the RL agent to learn how to reach different target locations with or without adaptable muscle actuators. And so we also, as part of this, had to implement a model of muscle adaptation. So we did this by implementing a function of use-based strengthening, basically muscle exercise, with kind of three rules. First, the RL agent is going to select the muscle activation intensity. Muscles that have stretched or produced force in a prior moment in time are going to experience adaptation and get stronger. And then muscles that get stronger and stronger, you don't want that to be unbounded, right? Biological muscle has its limits. So we also set an arbitrary threshold that our muscles can't get more than twice as strong. And we connected this muscle model with the RL agent and our structure, and we tasked the robot to learn to reach for this green ball. So as learning kind of episodes progress, we can see the robot get closer and closer to the goal until ultimately it does a pretty good job of reaching towards the target. But how does that muscle adaptation play in? Well, what we found is that if you try to design this biohybrid robot and you don't take adaptability into account, it can learn a policy towards the goal, but it's limited by the initial state of those actuators. If you allow the muscles to adapt like they would by having exercise in a real world setting, we're able to get better performance out of our robots. And most importantly, this adaptation did not negatively impact the ability of the reinforcement learning approach to find a control policy. And so this points towards some advantages that may exist in using these types of actuators for biohybrid robots. But how would you actually fabricate these crazy things, right? So there's a lot of kind of preliminary work that has to be done for biohybrid robots to kind of really come to fruition, right? So you're seeing pieces of it so far, and one of these pieces is manufacturing. Towards this goal, we're collaborating with Dr. Adam Feinberg in BME to build on the fresh printing platform. In this fresh printing approach, his group developed this technique. You're able to support really soft, fragile hydrogels in a bath while you're printing them. And that allows you to print really small parts and parts with internal cavities and overhangs. And this method is compatible with a wide range of biological materials, which makes it very appropriate for biohybrid robot fabrication. But of course, when you print with biological materials, they're very soft, they're very squishy, they're very fragile, just like biological muscle. And this poses a challenge for robotic applications where we want to be able to connect our muscle actuators to robots without them breaking. Therefore, we need reinforcement. In order to structurally reinforce our muscles, we've created some new bioprinting extruders in order to fabricate or to embed prefabricated fibers in hydrogels during our printing process. This long fiber extruder is composed of kind of two guiding tubes, and they direct the fibers through these wheels that then drive them into the bath and extrude this fiber. And the process is basically that you can print your hydrogel ink or your muscle ink in the future. You can embed these structurally reinforcing fibers and print more ink. And then to get the parts out of the support bath, you just have to change the temperature, and then you can have part retrieval. One of the primary benefits we anticipated from this type of printing was the ability to structurally reinforce our printed hydrogels. And to test this, we conducted tensile testing of alginate hydrogels as a stand-in for muscle with and without the fiber reinforcement. We found that, as expected, reinforcing super soft, squishy things with fibers definitely increases the ultimate strength and tensile modulus of our components, and that this was true both in kind of extension and in bending. And so this gives us a really good platform for then embedding a wide range of fibers into a wide range of hydrogels. So we've tested this a bunch with alginates derived from seaweeds, as well as collagen, which is a primary component in muscle inks. And we've tested this with a wide range of threads. So we've done silks and polyesters, threads made of collagen, little tiny wires. So it's a pretty versatile technique. We can embed single fibers or multiple fibers, as well as embedding fibers in 3D. So we've been mostly using alginate as a muscle mimic, but more recently, we're starting to print with actual muscle directly. And our ultimate goal is to kind of combine these techniques in order to put this fiber reinforcement approach into our muscle bioprinting. Hopefully I'm not running out of time yet, but I do also want to share a little bit about an alternative to biohybrid robots. With biohybrid robots, because you have these living materials, you have to keep them alive. So they have to function in very specific conditions and environments. And sometimes that can be a challenge, especially if you want to deploy these beyond the lab. So as an alternative, we're working towards creating sustainable biodegradable robots using plant-derived materials, in particular working with alginates and other marine-derived material components. These alginates have many of the same challenges as muscle. They're very soft and fragile. And so to overcome these limitations, we are again using the kind of embedded printing, fresh printing approaches in order to fabricate soft hydraulic actuators out of this bioink derived from common seaweeds. And we first validated this approach by fabricating and testing a number of small scale actuators, including bending actuators and linear actuators. And so this blue shape here, that is our alginate actuator. And just like any other kind of hydraulic or pneumatic robotic actuator, we can pressurize the component here at the end in order to get a transformation of motion. And the scale bars here are, I think I say 2 to 5 millimeters. I should remember that number off the top of my head, but these are very small and very squishy. And yet, they show relatively robust performance over hundreds of cycles. Some of them we've now taken out to tens of thousands of cycles, and they can maintain their functionality over that time. We can also use this approach to print a wide range of other robotic components. For example, in this case, we've printed a number of little soft robotic grippers. And actuation of the gripper then opens and closes the fingers, which allows us to manipulate small, rough, fragile, and slippery objects in these marine aqueous environments. And from the long-term goal of biodegradable robots, these actuators have strong potential for applications in marine ecosystems where traditional materials may pose hazards upon deployment or failure. So we've tested the degradation of these actuators in our sea slug tanks. Advantage of doing lots of different types of research. I also have marine aquariums in the lab. And we found that they were safely biodegradable in these marine reef environments. They're also even edible and digestible by marine invertebrates. And we can kind of program the degradation of these devices through chelation if rapid destruction is needed. And while there's still a number of limitations, we are then using these to push the boundaries of biodegradable and soft actuators. So where are we headed next? Well, there's a lot of ongoing challenges left in this field. And so just some of the key ones we're focusing on include packaging, life support, improving the force capabilities of our actuators, designing and programming these living neural controllers, and the development of better design tools for robots using these biologically sourced materials. And ultimately, we want to bring these capabilities together to create autonomous biodegradable robots. So I'll just wrap up by acknowledging again the wonderful students behind this work, my amazing collaborators, and our funding agencies that have supported all of this research and made it possible. And I'd be happy to take any questions. Thank you.
Thank you, Vickie. We very much appreciate all of these great insights that you've shared with us today. We will now turn to our audience for questions. As a reminder, please submit your questions via the Q&A feature at the bottom of your screen. Make sure to include your name with your question. So let's start with a question we got really early on. This is from Joanne Michelle Martin. Regarding the biological that is modeled, does the biologic have a learning period that can be observed and used to train your replica?
A learning period that can be observed. So a lot of the systems we're working with do have either learning or adaptation that they're experiencing. So we have not studied the learning in sea slugs directly ourselves, but the sea slugs were actually a model animal for learning research way back in the day. One of the Nobel Prizes associated with learning was done in sea slugs. The effect that might have on our biomechanical models would be that they can adapt and change their behavior over time, and we don't yet incorporate that into the models. So that can be one source of variability with the animal that would be really interesting to look at. The biohybrid robots could potentially also have adaptation. I'm not an expert in learning research, so I'm not sure that at the stage we're at, it would rise to what you would consider learning, but certainly the muscles can get stronger. You can change the strength of synapses between neurons, and that could be something that would be really interesting to study in the future for robot control.
Wonderful, thank you. Here is a question from Chad McMahill. In the earlier images showing the robotic learning through incorporation of adaptation, it appears that the robot never actually reached the object. Was that just a factor of this presentation, or was it prohibited from actually reaching the intended goal?
So we deliberately picked goal locations inside and outside of the robot's actual workspace. So I just, for the purposes of timing in the presentation, only showed the ones it had to really work for. The ones that were closer to the robot, both the adaptable and non-adaptable worms were able to reach those. But if we put the goal locations further away from the robot, and the locations were actually selected so that the non-adaptable one, the muscles just weren't strong enough to get the worm to the target. And what we found is that it would reach kind of the maximum extent of its workspace and then not be able to get closer. But if the muscles were able to exercise and get stronger, then over time it would get closer and closer to that goal location.
Very interesting. So we have a comment of, this is a very fascinating and excellent presentation.
Thank you.
Could these autonomous robots ultimately be able to communicate and collaborate with each other, possibly using agentic AI based on foundation models?
Ooh. Yeah, I'm not an expert in foundation models, but ultimately, yes, what I would love to move towards with this field is the ability to deploy many robots in an environment without having to worry about what happens if the robots get lost or broken, or since a lot of our applications are in marine and agricultural spaces, like what if the robot gets eaten, right? And you would potentially want to be able to deploy large numbers of robots in this space to do tasks like monitoring, treatment of crops, treatment of coral reefs. And then in those cases, the robots would definitely need to be able to communicate with each other. There are a lot of challenges with onboard electronics being biodegradable. So an area that we're thinking about right now is kind of this line between make the components that can be biodegradable, make the other components recyclable so they can be used across multiple robots. And then we would have to think about what can you package into that volume to allow communication and coordination, and probably some cloud connectivity if you wanted to connect it with some sort of foundation model.
Well, your answer kind of rolls right into this next question. So what are some of the benefits of the components being biodegradable?
Yeah, so I see this as having a couple key contributions when we're able to achieve these in deployable robots. The first one, like I mentioned, is being able to release a swarm of robots. You know their degradation profiles, so they can go complete the mission you want them to do and then degrade naturally. You don't need to expend additional resources to go recollect them, and they're not going to damage the environment. So that's one application area. Another is potentially in the space of just needing to be able to reduce single-use components. So a lot of soft robotics is adjacent to like wearables, right? So wearable robotics, exoskeletons, and those have right now a lot of single-use components. So if we can replace the single-use part with something that's biodegradable, then that minimizes the waste of those types of devices. Or for agricultural applications, if you want to be able to do monitoring in fisheries, fish are not very bright. If you put a lot of little tiny fish or little tiny robots in a fishery, chances are somebody's going to try to eat a robot. So you don't want them to be dangerous to the organisms in the environment.
Absolutely. So these two questions I think are kind of related. So I'll just give you them both. The first was, can the components be self-healing? And the other are do the muscles have self-growth capability in some environment? So I feel like those are pretty closely related questions. So I thought I'd.
Yes, yeah, absolutely. Self-healing is something we're really interested in right now. And there's kind of two areas of self-healing that we have to think about. One is just almost more self-sealing, right? So can it reconnect so that force can be transmitted across the break again? And our current biohybrid actuators should be able to do that. We're also collaborating with some other folks in MECI here, for example, Carmel Majidi, who does a lot of work in soft electronic skins. And there we're really interested in, can you use like a self-sealing skin to kind of package these systems? So there's that aspect of it. And then there's if it's a substantial damage to the actuator, could the use of living materials help you overcome that? I think the long-term answer is probably, but there's a lot of foundational research that needs to happen to get us there. right now, we're working with muscle actuators using myoblasts up through kind of matured muscle. And so we don't have any of what are called satellite cells. In our bodies, we have these locations with these satellite stem cells that can differentiate and become muscle, but aren't muscle yet. And they're kind of in waiting for us to accidentally injure ourselves so they can go migrate to that injury and repair it. And since we don't have that cell niche in our muscles, there's going to be a limit to the type of injury that they could repair from. But they might be more repairable. So you might be able to envision a future where these biohybrid muscle-based actuators, it gets damaged to a level that it can't overcome, and then you just bring it back into the shop, essentially squirt in more muscle and let it kind of repair that way. So these are all things we're thinking about, but they're not there yet.
Very interesting. So with consideration of being biologic with robotics, what physical environment limitations are currently in place, like atmosphere, food?
Yeah. So right now, all the biohybrid robots in the lab live in sterile cell culture incubators. They only get handled in sterile cell culture safety hoods. They have to live in basically a petri dish full of a medium that has the right salt balance, the right pH, has glucose for energy, and a graduate student or undergraduate researcher has to go in there and feed them regularly and change out that liquid. So that's a big constraint on deployability of these devices. And it's a really active area of research in our group right now. How can you package these to get them out of that environment? How can you incorporate systems to provide them with more glucose or oxygen or process the waste that the muscle produces? And so John mentioned this ARO Muri that we're leading, and that's the key focus of that work, because it is a really big challenge right now.
So can you talk about a little bit about some of the application areas, and then will there be any application areas in artificial human tissue and organs?
Yeah, so talked a little bit about the environmental ones. So kind of focusing on possible applications in more of the medical adjacent space. Absolutely. So biohybrid robotics uses essentially all of the same fabrication techniques as tissue engineering. And we really build off of the tissue engineering literature as well. So there are biohybrid groups that are looking at, if we build this muscle actuator and we can control it and get it stronger, could that then be an implant, right, for things like volumetric muscle loss repair? There's also long-term, long-term, like many tens of years, visions of building on these techniques, scaling up these actuators. And if you can get them from the little tiny scale that we're currently building robots to a more human scale, then we can start talking about biohybrid prosthetics. And that could be a way to bring this back into that kind of medical space as well.
That's great. Okay, we have a few more questions here. As muscles get stronger by absorbing proteins, how would it work in biological robots? Would they need to feed like a biological creature?
Yep. Yes. So yes, it's I can speak from experience when I was a grad student, and I'm sure my current graduate students and postdocs feel this as well. It's a very unique challenge to be a robotics researcher, where if you don't go regularly feed your robots, they die, right? So we do have to take care of them. They require new food about every other day in the lab. And that's going to be something that when we deploy these systems, we have to think about. Either you need to be designing biohybrid robots for missions where the mission is short enough that they can go do the thing, come back and get more food, or we need mechanisms for them to be able to get food from the environment. And I don't have solutions for that yet, but it'd be very interesting to look at.
Yes, absolutely. So we have another question from Chad, who asked a question earlier. in the restriction of limiting to no more than 2 times the previous strength, what time delay is put in place? It seems like they could reach a super strong condition if that multiplication periodicity were too short, such as microbursts to support significant strength increase.
Yeah, so all really great questions. The parameters for how rapidly they were allowed to get stronger and that threshold in that preliminary study, those are those were picked kind of Edisonianly, right? So that was experimentally tuned based on the grad students on that project having looked at the literature and built their own muscle actuators in the past, trying to be reasonable, but the data just doesn't exist to really tune this to our specific actuators yet. So one of the preprint for that paper is up on arXiv if you want to check it out. But one of our goals right now is actually I've got postdocs fabricating actuators, and we're going to get the data that we need to actually tune those hyperparameters to our muscle. And that'll make that design tool even stronger for us to then be able to build these robots.
Great, thank you. Do you envision, this is from Thomas Kirk, do you envision the hybrid muscles ultimately being part of the human body to be controlled and sustained by the nervous system and bloodstreams?
If we can get the scale up to the point where these could be biohybrid prosthetics, that's where it almost, the line between robot and like engineered organ starts to blur, right? And if we can scale these up, then we can start looking at, these are almost more replacement muscles, right? For injuries, for, replacing tissues after large resections and surgery. And in those cases, if you've got that large of a muscle, you would, I think, ultimately, yes, want to integrate it back just like it was an organ, right? It would almost be like doing a kidney replacement. And that has a lot of its own challenges. Nerve regrowth and nerve reinnervation is an active area of research all of its own. But I think hopefully by working kind of on these research thrusts in parallel with biohybrid robotics folks really trying to maximize the force capabilities, look at scaling up tissues, look at these interfaces so that we can actually like reconnect these tissues to structures, and all of the great research that's going on in tissue engineering and in the biomedical research space, that these will drive us towards that as a future vision. In the long term.
Great. So shifting gears just a bit, you mentioned earlier that undergraduates participate in your lab, undergraduate students. What do they have to do to participate? Do they have to take a specific class, or is this something you have to interview for? How does that process work?
Yeah, so it depends a lot on the specific project that a student is interested in. As you saw, the group's gotten pretty big at this point. I love having a big lab. I love having students collaborating with each other. And so what we have found to work really well for us is I let my senior PhD students and my postdocs build their own little research teams. And so we'll work together to identify like the scope of a project that might be able to support an undergraduate researcher or a new master's student. And then we advertise those. So the MECI department does a really great job of posting project opportunities, both to our master students and to our undergraduate students. And so the students, the undergrads that are interested will usually like send me a resume and tell me which project topic they got excited about. And then I'll share that with a PhD student who sets up an interview and sees if there's a good fit. Or I've had students just, stop by and ask about opportunities. Most of the time when they do that, I tell them, yeah, send me a resume. I'll forward it to my PhD students. I don't have specific requirements as far as like, you must have taken this course. We do such a wide range of research in the lab that there's a lot of opportunities for people at kind of different academic levels. And it more comes down to, is there a good interest fit and at least some background knowledge related to the project opportunities that we have from the graduate students. So we've had people start as freshmen. That's a little harder just because they haven't had if they're a BME student, they haven't had like BME lab yet, or they haven't had dynamics yet or statics. But there are still ways to get people involved that early. And then we've also had just a lot of folks kind of come in sophomore, junior year, even a couple folks just come in as seniors.
Great. And then I'm going to just take one last question before we wrapped up. So this is from Chad again, and he said, other than the described and continued need to give sustenance, what appears to be the issues of concern for upscaling in size and interaction between individual bodies?
Oh, I mean, sustenance is a big one. A lot of our actuators right now are small so that we're below the diffusion limit of oxygen. So if you grow a tissue that is about no more than like 500 micrometers in diameter, oxygen and glucose can just from diffusion get all the way through the tissue so that the center of the tissue stays alive. But if you get bigger than that, the cells in the center can't stay alive. So that's a big limit. To go above that, we're going to need to do vascularization in order to keep bigger tissues alive, which has its own challenges. So yeah, sustenance, a lot of it comes back to sustenance. Another big challenge from the robotic standpoint is those interfaces. Muscle is about 30 kPa, which just kind of means it's about like Jell-O. So if you imagine like trying to put a strip of Jell-O on a pole and pulling on it, the Jell-O is going to tear. And our body gets around this by having that muscle tendon bone junction. So we've got a mechanical gradient. So it is nice and strong and it's not going to just tear off of the bone. We don't have that yet really for biohybrid actuators. And so that's something that we're looking at as well, is like, how do you do those interfaces, both from like that mechanical standpoint, but then also from a control standpoint, how do you do the electric interfaces? So yeah, sustenance and interfaces, I think are probably my two favorites if I had to pick two challenges.
Great. Well, thank you again, Vickie, for a very thought-provoking and informative program. I would like to also thank our audience for participating today. I hope you will be able to join us at our future Faculty Insights programs. Please keep an eye out for a follow-up email soon, which will include a link to a recording from this session. Thank you, everyone, again, and have a great day.